Good morning, everyone, and welcome to day 2 of the 34th edition of Soup and Science. My name is Laura Nilsson, and I'm the Associate Dean of Graduate Education for the Faculty of Science. I'm also a professor in the biology department, and today I'm joining you from my lab over here in the corner of campus in the beautiful Stewart Biology Building. Before we begin, we acknowledge that McGill University is on land which long served as a site of meeting and exchange amongst Indigenous peoples, including the Anishinaabe and Haudenosaunee nations. We acknowledge and thank the diverse Indigenous people whose footsteps have marked this territory on which peoples of the world now gather. We are joined today by 5 wonderful speakers who will tell us about their research. So we'll jump right in and get started with Professor Eric Glaciologic from the Department of Mathematics and Statistics. Welcome, Eric. Go ahead.
Thanks, Laura. Just share screen here. Dive right in. Okay, can you see that?
Yes.
Great. Okay. All right, so my elevator pitch here is around what would you do with a network, right? We have networks all around us now, biological networks, social networks, transportation networks, and they are, for someone who works in mathematics and statistics or engineering or other fields, they are a data object that is interesting to study. To motivate why it's not just the same old, let's actually start with the same old, right? So here's something that you would get from a statistics 101 type course, comparing 2 samples. You might have those identifying female responding with a certain set of numbers, those identifying male responding with another set, and perhaps you've been taught that you then take an average and you can compare averages to begin gaining insight into whether there's differences in the groups or whatnot, right? We often then try and talk about the uncertainty associated with whether or not there really is a difference. If I got different numbers and they were slightly different, does that mean that there's just a difference in my data, or does it mean there's really a difference in populations? That comes from adding probabilistic infrastructure to it. So you learn about normal probability densities, or perhaps even multivariate normal or things like that. And with these types of shapes, you start adding probabilities, like the probability that there is a difference is this, or the probability I would see such an extreme difference is such and such, right? And from that, we really build outward, right? And so all of the statistics foundations, a lot of what we do in machine learning and related areas are really based on kind of building up from a data type, understanding its structure, putting probabilistic statements around it allows us to do estimation. We can make decisions. We can do predictions of the future. Where my research comes in is with this question, right? What if the data point, quote unquote, is actually a whole network? And I like to use this example from work that I've done with colleagues in neuroscience over a decade or more, where we work very hard to get large amounts of data that after we process them using very sophisticated methods that were their own research at one point in time, we end up with a network. It's so that we can do this for thousands of people. So now we have a data set full of 1,000 different networks. But how do you analyze things that are a bunch of points in line? And so I'll leave that there as sort of the point of questions or whatnot. But that is basically what the essence of the research about. Points and lines aren't really geometric. Points and lines aren't really like tabular. But there are ways that we can do all the things that I talked about and more. And a lot of that's been what we've been developing over the last 10 years.
Thank you, Eric, for that very interesting and stimulating talk. A great elevator pitch. Next, we have Professor Nicolas Moitessier from the Department of Chemistry. Go ahead, Nick.
Hi, everyone. So I'm Nick Moitessier from Chemistry. I'm going to tell you a bit of a story of the way we'd like to see chemistry being done, or the way we envision chemistry and how it's going to be done in the future. All right, so let me give you or tell you a little story. So what you see here is glucose, basically the sugar that you put in your coffee on the morning. And what it does when you swallow this saccharose, it will at some point reach a protein. I mean, this going to bind to this enzyme, and what this enzyme will do, it's going to break saccharose into glucose and fructose. And then the enzyme is released and going to be able to process another molecule of saccharose. And it can be doing this on and on and on and on. As long as you're eating sugar, you're going to end up having a lot of fructose and glucose in your bloodstream to be used by your brain, your muscles, and so on and so forth. All right, so that's what we call an enzymatic process. Now let's say this enzyme is a different one processing saccharose, is going to be processing something else that may induce a disease. So I want to develop a drug that basically kills that enzyme, because this new enzyme now is involved in cancer progression, is involved in Alzheimer's disease. So my job as a drug developer, as it can be called, is to design a molecule that's going to block this enzyme. So the strategy is very simple. We're going to take a molecule that looks like the substrate, initially was saccharose, except that this drug cannot be cleaved. So now it's going to bind, and at this stage, the molecule cannot be processed by the enzyme, and the enzyme is dead. So developing a drug is basically trying to mimic the natural substrate of this enzyme, and that's our job. So as I want to develop a drug binding an enzyme, killing an enzyme, I need to develop this molecule. How do we do it? The main problem in this process is that to have a drug being active, you need not only this molecule to block the enzyme, but also to be non-toxic. So I can tell you that Tylenol is killing Canadians every year. No side effects. Aspirin is known for its many side effects. No resistance over time. And obviously resistance to antibiotics is well known and well described. So there's a lot more than just getting an activity or a blockade of an enzyme. And then you have a lot more. It's going to be metabolized in your liver. It has to be selective. It has to be chemically stable. You know, you're going to store it on your shelves in your bathroom, so it should be there and still be there months later. Should not be binding too much to protein in your plasma. It should be synthetically feasible, so you need to be able to make it at the ton scale, and so on and so forth. So there's a lot of work to develop a drug, and having all of these properties checked. So obviously, you can be lucky. A big pharma would not rely on this approach too much, but many compounds have been developed using luck. The fun fact, Viagra has been developed to be treating heart diseases. And you know, the activity that it's treating now is clearly not the same. You can screen at random millions of molecules, hoping that one is active. You can do some more rational design, and hopefully you can be using software that would do all of this virtually. And why do we want this? Quickly, 15 years to find a molecule that is checking all of these boxes. And I really like this statement. It's much harder than rocket science. So medicinal chemistry is harder than rocket science, simply because, you know, you look at this, 12 years before, between the start of the space age and the man on the moon. And as soon as you have one rocket, you can make another copy of it, and a third one, and the fourth one. The problem is every disease is different. So you have to start the process over every single time. And it costs billions to come up with a drug and to read. So we need to find strategies, and you can do large screening, so randomly trying to find a molecule. You can do rational design, where you try to find the properties and how a molecule can act in the way you want it to act, or you can do virtual screening, so you're testing everything on computers. What does that mean? That means that you're basically going to give millions of molecules to your computer and say, give me one that is active, non-toxic, soluble, doesn't bind to plasma protein, is metabolized by the liver properly, is excreted by the kidneys, and then it would tell you which one to make, and you're going to stick in a short period of time, you should be able to have a drug. In much shorter than the 15 years that we need now. So our job in our lab is to develop all of this software to test it in real scenarios. We, for instance, develop molecules that are apparently effective in Parkinson's disease, at least in living neurons. It clears the protein that is the bad protein in Parkinson's disease. All of this predicted by software. And the idea is really to predict all of these properties and in a short period of time, so it would be a lot faster than doing all of the experimental work. And that's pretty much what we do. Thank you.
Thanks to you for that very interesting talk. We'll move right ahead to our next speaker, who is Professor Pia Wintermark from the Department of Pediatrics. Over to you, Pia.
Sorry, I was muted. Okay, that's better, probably. So I just wanted to discuss what we do in my lab. As I mentioned, I'm a neonatologist, so I do intensive care for babies, and I also have a lab. In my lab, we try to understand the cause and the consequence of brain injury in sick babies. And we try to develop and test strategy to prevent brain injury. And we test some medication like not as thorough as the previous speaker. So, and we try to improve the future of these babies. So we have several parts in the lab. We do clinical research. We use brain and magnetic resonance imaging and bedside monitoring to study how the injury in the baby develop. We have a basic lab where we have a rat model of the same disease in the baby, and we try to test the treatment to modify the brain injury. And then last, we are now running a randomized trial where we are trying one of these medication and see if it works in the baby after the promising result we got in rats. So we are running the trial in Canada and in Uganda. One of the medication we tested up here on the slide of the previous speaker is sildenafil, also called Viagra. It's an inhibitor of the phosphodiesterase type 5. It's more known for other effects in the body, but in the brain has several effects that are less known. It can act on neuron and favor the growth of new neuron and synaptic plasticity. It can remove neuroinflammation, can promote myelination, and it also acts on the vessel. And with all that, we think it may repair the injured brain of babies. I want to show, we had a summary of what previous students have done in the lab, undergraduate students. I had 27 come to the lab in the last 10 years. Some of them, as you see, have received some award. Some of them have been co-author on abstract, and some of them have been to local conference. Some of them have been co-author on manuscript, and 3 have been first author, and some of them have received prize. So if you come to the lab, we'll make you participate to something that we are doing. This is usually the course for which I can take students, because I'm not directly in the Department of Science, but Faculty of Medicine. And this is the way to reach me if you're ever interested to come to the lab. Thank you.
Thanks for that great talk, Pia, and thanks for the reminder of undergraduate involvement in these research programs and how undergrads can go about taking that step from hearing about interesting research at Soup and Science and really getting involved in research. There's a lot of different opportunities for undergrads to do research in the Faculty of Science. It's something that we really pride ourselves on. There are summer scholarships that support students to work in labs. And as Pia showed on her nice list of opportunities, there are different ways to do research for course credit. So really important to stay informed about. We'll do the first will be Yuyan Chen from the School of Computer Science. Go ahead, Yuyan.
Okay. Hi, everyone. My name is Yuyan, and I'm in my 4th year in Math and Computer Science. I'm going to talk about modeling calcium dynamics in astrocytes. So first, what is an astrocyte? Astrocytes are star-shaped glial cells in the central nervous system and have many important functions like regulating blood flow, transferring mitochondria to neurons, and supplying the building blocks of neurotransmitters. And where is calcium stored inside an astrocyte? There are 2 internal compartments. The first is endoplasmic reticulum, or for short, ER. They're the primary driver of intracellular calcium fluxes. And there are also 2 major types of calcium channels, IPR and RYR, that are on the membrane of the ER. It's mitochondria. Mitochondria is a major internal calcium store, and they form microdomains of high calcium concentrations. So there are 3 types of calcium signals in the astrocyte. The first is blips. Blips are weak and brief signals. They're from the opening of a single receptor. And the other one is puffs. Puffs are higher in amplitude and duration, and they're from the opening of a cluster of receptors. And finally, you have waves. Waves are from the propagation of calcium puffs from one cluster to another via calcium-induced calcium release, which is a very important mechanism for calcium dynamics. And now to model the calcium propagation, we consider the calcium source, like over here, the circle over here, as a moving front. And we think their diffusion is anisotropic, which means their diffusion depends on both the position and the direction. And this is our guiding partial differential equation. So C is the calcium source, T is time, V is the velocity, and N is the outward normal, which basically means it's expanding. And the velocity is related to the distance to boundary. And our biological motivation for the diffusion speed is that we expect the calcium to have more isotropic diffusion in the soma, or the cell body over here, and we expect them to have more anisotropic behavior, so they will diffuse faster along the same process over here and slower across the process. And now I have 2 videos to show the result. Oops. Yeah, so here we have a single source of calcium, and it's gradually diffuses to the soma or the cell body over here. And in the next video, we have multiple calcium sources, and then finally diffuse to the entire cell. And oops, yeah, that's it from my presentation. Thank you.
And thanks to you, Yuyan. We'll move right along to our next speaker, Ruth Wang, who is from the Department of Physics. Go ahead, Ruth.
Hello, everyone. I'm Ruth from Department of Physics. This summer, I was working with Professor David Cook in our terahertz optics labs. Our lab played with terahertz light pulses. This is a cool picture of laser I found online. And what are terahertz pulses? Terahertz pulses are light pulses of size between microwaves and infrared. Their spatial size is in the unit of microns, which is 1 magnitude down up to human here. So they're really big. My project is to numerically simulate experiments on a relativistic moving mirror. What is a mirror? It can be anywhere with a sudden increase on refraction index. We can realize that by exciting a dielectric medium. Here is a cool picture of the dielectric medium. And if we send it up a compound, we excite the medium from 1 reflected at the front. In that way, we create a mirror. Then we send in a second terahertz pulse, which will be our light pulse. Some pulse will be reflected, the rest will enter the transmitted field. Now we have our mirror. How do we speed up to make it moving? Can an object travel faster than the speed of light? Of course not, but it all depends on what velocity we are talking about here. Here, the relativistic moving front does not carry any information. You can think of as scissors or two blades dropping on each other. So you can model it by a boundary tangent function. You can even accelerate to infinite speed. Here is a 3D space-time diagram showing the reflection of the mirror. You can see our pulse got reflected. These are dispersion diagrams describing the reflection of the pulses as well. On the y-axis is the frequency, and on the x-axis is a wave number. You can see we pump the pulse from the red dot to the black dot. And here we can accelerate it at any random speed, infinite speed, or zero speed. These are different cases we investigate. Interestingly, we have case 3, which we trap call it the optical push broom effect. And in case 4, we stop the pulse because they have zero group velocity. And in case 5, we reverse the time for the pulse. And what's in the future? Our next step is to compare accelerated moving front with constant velocity case. Is acceleration just different constant velocity at different time moment? And we are making a 2D simulation, which will look like a puddle in a pond. Maybe we can pull something out of the quantum vacuum, but that would be really cool. We don't know if it's realistic. And the physics we are doing is analogous to the physics of Hawking or Unruh radiation, because we are dealing with relativistic physics. And that's all, and I'll take questions in the breakout room. Thank you.